aspirants. So here we are starting out the major chapters, that is genetics, where the first chapter from here involves principles of inheritance and variation, where we are going to start with this. First, let us discuss what are the major concepts that we have to cover from this chapter. Okay, from principles of inheritance and variation, firstly you have to be clear with the terminology that we are using in this chapter, followed by one gene inheritance and exceptions of one gene inheritance. Okay, one gene inheritance and exceptions of one gene inheritance. From monohybrid cross, Mendel has given out two major laws. Those are law of dominance and law of segregation. What are the deviations from that laws we will learn. Under exceptions of Mendelian's principles from monohybrid cross, we will learn the concepts like pleiotropy, incomplete dominance, codominance, multiple allelism. These are the concepts we are going to learn today. Along with this, we will also learn about Mendel's dihybrid cross, that is two gene pattern of inheritance, followed by polygenic inheritance we will be learning. And the next set of this chapter includes the concepts like chromosomal theory of inheritance by Sutton and Boveri, followed by the experimental proof for chromosomal theory of inheritance given by Thomas Hunt Morgan, and then mutations, which is very important, and then Mendelian disorders and chromosomal disorders we will be learning. At last, pedigree analysis and sex determination. These are the concepts that we are going to cover from this chapter, and they are very, very important, not just for your EAPCET but also for your NEET. Okay, so let's get started first with the major terminology that we are using in this chapter. First is inheritance and variation. So here you see inheritance and variation. What is inheritance? Inheritance is the characters that are being transferred or transmitted from one generation to the successive generation is known as inheritance. Okay, transfer of characters or traits from parental generation to the next successive generations is called as inheritance. What is variation? The degree by which the offspring differs from the parent. Okay, of course will be same like that of our parent. Most of the traits or characters will be similar, but then there are major differences. Okay, those are addressed as variations. Okay, what is variation? The degree by which offspring differs from parents. And next is character or trait. Any physical appearance, any character, okay, any physical appearance is addressed as character, which is phenotype. If it is transmitted to the next generation, for example, hair color is black is one character, okay, and eye color, skin color, height, all these are characters. For example, tall height is transferred to the next generation, then that character is addressed as trait. Okay, whichever characters are transferred to the next generations, those are addressed as traits. Next is what factor. Factor is the term used by Mendel. Mendel used the term factor. What does it mean? Okay, what does factor means? Factor means like every character or trait is coded by one factor. These factor occurs in pairs. These factors occurs in pairs. Where are these factors present? Over DNA. Okay, let us assume the double-stranded DNA. Okay, double-stranded DNA in a nucleus. Let us assume this is a nucleus and a double-stranded DNA. Okay, those factors are present in two successive chromosomes, in homologous chromosomes. These two are homologous chromosomes. For example, let us assume height only. Height wherein capital T and small t. Here one chromosome coding for capital T, another for small t. Okay. These are addressed as homologous chromosomes because two alternating forms of a factor are present at the same loci of these two chromosomes. Okay. They are present at the same location or loci of the two chromosomes. These are known as homologous chromosomes, and these are two alternating forms or contrasting forms of a single factor, that is known as alleles. These are known as alleles. Alleles occur in pairs, okay, and the pair of alleles is factor. Clear right now? So this is about factor or gene. And then if you observe, next is alleles. Alleles, if you see, these are two alternating forms of which one will be dominant and other will be recessive, followed by homozygous and heterozygous chromosomes. Homozygous chromosomes wherein if both the forms, both the alleles are of same type, be it either capital T or small t. This is homozygous nature of factors or homozygous chromosomes we call it as. Heterozygous nature are two alternating forms of allele on a chromosome or two alternating forms of allele on a gene. Clear, right? And then phenotype is the appearance, physical appearance that is being easily noted out. Genotype is the genetic expression of an individual. The genetic expression of a trait is genotype. The physical expression of a trait is known as phenotype. I hope all the terminologies we are using here is clear to you. Both homozygous and heterozygous chromosomes, you have to be very clear, because homozygous chromosomes we have already learned in cell division, where homozygous chromosomes and heterozygous chromosomes we have learnt it already. Okay, homozygous genes and heterozygous, capital T, small t. Fine, right? Yeah. Those chromosomes which are exhibiting two different forms of alleles at same locus are known as homologous chromosomes. Sorry, homologous, not homozygous, it is homologous. Okay, homologous chromosomes. Non-homologous chromosomes means if the alleles are at different locations, then you call it as non-homologous chromosomes. Homologous and non-homologous, remember that. Homozygous, heterozygous is for genes. Okay, if the genes are having two same alleles, tall or dwarf, same alleles are present, that is homozygous. Two alternating forms, it is heterozygous. Clear till here? Any doubts anywhere, ma? And then so even before Mendel, way back around 8000 to 1000 BC, human beings have known the importance of artificial breeding, okay, and the variation which is involved in sexual reproduction. Humans are aware of that, because of which the hybrid varieties in bulls and cows were produced, like Sahiwal cow of Punjab and Ongol bulls. Okay, these are the examples which are given in your text, you can go through it. And it is Mendel who has worked for 7 long years on garden pea, that is Pisum sativum. It is important for us in statement based questions on how, on why Mendel has chosen Pisum sativum. So here if you see, it is Gregor John Mendel who has worked for 7 long years, 7 years from around 1856 to 1863. Okay, for 7 years Mendel has worked on Pisum sativum. Why did he choose to garden pea is the question? Because garden pea is an annual plant, it completes its life cycle in a single year, and it exhibits clear contrasting characters, be it in the height. As such, 7 contrasting characters were taken into consideration by Mendel, okay, be it seed color, seed shape, pod color, pod shape, flower color, plant height and flower position. These are the 7 traits which are considered by Mendel. And self-pollination will give rise to true breeding lines in pea plant, okay. Continuous self-pollination of garden peas will give you out true breeding lines. What are true breeding lines? True breeding lines are nothing but exhibiting homozygous nature, be it for dominant or recessive. Homozygous nature is exhibited and stable pattern of inheritance is shown. Stably the traits are being passed and they are present in homozygous nature, okay, for a stable pattern of inheritance. When a single trait is crossed across many successive generations, in many generations, when a single trait is crossed, okay, and that exhibits true breeding line, or after continuous self-pollination it exhibits true breeding line which shows stable pattern of inheritance of homozygous nature. Clear? And then artificial cross-pollination. Artificial cross-pollination is also easily done here by dusting the pollen grain of the desirable interest and stable pattern of inheritance is shown. So, what are the major features that Mendel has chosen Pisum plant? What are the major reasons why he has chosen the Pisum sativum? 1. It is an annual plant. 2. Clear contrasting characters are shown. 3. Self-pollination, and then artificial cross-pollination, stable trait of inheritance is shown. So, these are the noted features. And then next we have flower color, seed shape, seed color, pod shape, plant height and flower position. Of all of the traits, you should be clear on which is dominant and which is recessive. If you have to consider flower color, dominant color is purple, recessive is white. So, this is dominant and this is recessive when flower color is considered. Seed shape, round is dominant seed shape and wrinkled is recessive seed shape, okay. And seed color, if you see, yellow seed color is dominant and green seed color is recessive, fine, right. And then pod color, green color is dominant and yellow color will be recessive. Pod shape, in flattened pod shape is dominant and constricted pod shape will be recessive. Plant height, tall plant is dominant and dwarf plant is short plant which is recessive. Axial flower position, that is flower position at axillary region is dominant and flower position at the terminal region is recessive, fine, right. So, these are dominant and recessive traits. Can you see? Dominant and recessive. For all the 7 characters or traits, you have to clearly remember like which is dominant and which is recessive. If you want, you can take a screenshot of this, because not every time the question will be put on the height, not every time the question is put on seed color. Any character is taken of which 2 traits of it will be given. You have to easily analyze it. And there is one more statement base, you have to be clear on that. Whenever the question is asked like how many characters or traits are considered by Mendel, you have to choose 7 characters or traits are considered. 7 characters or traits are considered. How many varieties of it? 14 varieties. How many varieties of it? 14 varieties. Fine, right. 7 characters or traits are considered, of which 14 varieties are noted. Clear till here? Any doubts anywhere, ma? And then artificial hybridization is done, how crossing is done. As pea plant is a bisexual plant, it has bisexual flowers, okay, it has bisexual flowers. In a bisexual flower, you have to first emasculate, or you have to cut down the anthers, so that it will prevent the self pollination. From a bisexual flower, what is the initial step we have to do? We have to cut the anthers. Then we have to bag up this flower. After emasculation, we have to bag up the flower with a polythene cover. Then the pollen grains of desired interest should be dusted on this stigma, okay, post which it have to rebag again. And then, for example, after emasculation and bagging, you will dust the pollen grains and rebag it. If at all you want a monohybrid cross, then you have to choose 2 flowers which are variant in single trait. If you are going for a dihybrid cross, then 2 flowers you have to choose which are showing, the 2 plants have to show variation in 2 traits. Trihybrid or tetrahybrid, any cross if you want, you have to look into the number of varieties that you are taking. 2 varieties, then it is monohybrid cross. 4, it is dihybrid. 6, it will be trihybrid cross. Clear? Understood? As such, after crossing and all, you will get the seeds, collection of the seeds, and proceeding for the next generation. This will be the process or the sequence of artificial hybridization. Clear, right? My emasculation, bagging, dusting of pollen grains. You can take or choose the flowers based on the number of variants. Monohybrid cross, 1 gene inheritance, dihybrid 2, trihybrid 3, okay, and varieties will be 2 here, 4 here and 6 here. Always be clear with the number of traits and number of varieties. In any cross it might be. And post fertilization, you can collect the seeds once after the production of seeds is done. This is the process of artificial hybridization. Now let us look briefly into the monohybrid cross, which is very clear. Monohybrid cross, which is given in our text, is the consideration of only one character or trait, that is plant height, of which we have tall and dwarf. Yeah, tall and dwarf. In first parental generation, remember students, all the time parental generation is taken with homozygous condition only. Parental generation is with homozygous condition only, homozygous tall and homozygous dwarf. In F1 generation, you can see all the plants obtained were tall plants only. F1 generation is known as first filial generation. After the crossing of homozygous parents, you will get the phenotypic ratio of F1 generation is completely tall plants only. Okay. Here itself I will explain you the test cross. Here if you see the phenotype, you can know that all the plants are tall. But you have to also know the genotype, whether it is having homozygous condition, the tallness is of homozygous tall or heterozygous tall. This is a big question mark, yes or no? F1 generation, all the plants obtained were tall, but it is a big question mark whether it is homozygous or heterozygous. Here comes the concept of test cross. What is test cross? The plant, here the plant whose genotype is unknown, that plant when it is crossed with the recessive parent, you can easily identify the genotype of that plant. Let us suppose all the plants obtained in F1 generation are homozygous tall, and when it is crossed with homozygous dwarf, that is recessive parent. Test cross means what? No? Crossing of the genotype of the plant which is unknown with its recessive parent. Here in simple terms I can say you crossing of F1 generation with the recessive parent to identify the genotype of that particular organism. So here if you see, all the plants obtained will be tall plants only, yes or no? All the plants obtained here will be tall plants itself, and they will be heterozygous tall. When all the plants obtained are of dominant phenotype after test cross, it is noted that the unknown genotype is homozygous. Understood my point, everyone? Here you see heterozygous tall. If F1 generation it is showing heterozygous tall, here you look into, that is capital T small t, heterozygous tall, when it is crossed with the recessive parent. Okay, here you see what you will get, capital T small t, and then so you will get two dominant and two recessive phenotypes in 1:1 ratio. So you will get two dominant and two recessive phenotypes in 1:1 ratio. So if at all, when the phenotype of when the genotype of the unknown plant crossed with the recessive parent, you will get, when you are getting one dominant and one recessive trait, then it is known as the genotype is heterozygous tall. Okay, the genotype is heterozygous tall. If it is homozygous tall, all the offsprings will be dominant phenotype only. If it is heterozygous tall, you will get 1:1 ratio. Clear till here? Any doubts anywhere, ma? Clear, right? And then in F2 generation, if you see, so here in F1 generation we have stopped here. We have understood clearly about test cross. Test cross is to know about the genotype of the F1 generation. If it is homozygous tall, all the offsprings obtained will be dominant phenotype only when you are crossing this with recessive parent. If it is heterozygous tall, when you cross it with your recessive parent, you will get 1:1 ratio of dominant and recessive traits. This is how you will identify the genotype of the F1 generation. In F2 generation, after crossing or self-pollination of F1 generation will give you the F2 generation. Self-pollination of F1 generation gives you the F2 generation. So in F2 generation, when you see, you can clearly note down the phenotypes. 3 will be dominant phenotype and 1 will be recessive phenotype. So the phenotypic ratio will be 3:1 and the genotypic ratio. How can you identify the genotypic ratio? Knowing from the test cross, the genotype is capital T small t. When capital T small t is selfed out, what you will get, ma? When capital T small t is selfed here, yeah, you will get 3 dominant phenotype and 1 recessive, and genotypic ratio 1:2:1. Clear, right? So from this monohybrid cross we have learnt what will be the parental generation, what will be the F1 generation. In F1 generation, to know the genotype, what we are doing, test cross, and in F2 generation, after obtaining the self-pollination products, we can easily identify the phenotypic and genotypic ratio. How can you identify this? By conducting the test cross of F1 generation only. When you get to know about the genotype, then only you can get to know about the genotypic ratio of F2 generation. Clear, right? Anywhere if you are facing any problem, please let me know. And then here this is given out in a Punnett square. What is a Punnett square? Punnett square is nothing but the graphical representation of maximum gametic fusions that can be exhibited in the time of fertilization. What is Punnett square? Punnett square it is the graphical representation which will show you the all possible crosses of the gametes. Okay, It is a graphical representation which shows all possible crosses of gametes during the time of fertilization. Clear till here? Any doubts anywhere, ma? So, based on the monohybrid cross, Mendel has given out two major laws, significant laws. Those are law of dominance and law of segregation. What does law of dominance say about? Law of dominance says that characters occurs in pairs. Characters or traits occur in pairs. Firstly, characters or traits are coded by factors and each factor occurs in pairs. In each pair, you can see two contrasting traits. Two contrasting traits where one allele will be dominant and the other allele will be recessive. They are coded by alleles, they occur in pairs and those are alleles. One will be dominant and one is recessive. And remember, law of dominance is not universal. Okay, characters coded by factors, factors occur in pairs and each pair will have one dominant allele and one recessive allele. Dominant allele will mask the effect of recessive allele. Remember that. And next is law of segregation. In a single factor or a gene, factor is now called as gene. In a single factor or gene, when it has two alternative forms, let us suppose capital T and small t, when they are present together in heterozygous condition, alleles doesn't mix or blend with each other. Okay, alleles doesn't mix or blend with each other. They exist in their unity. Okay. And remember, law of segregation is universal. Clear, right? Law of dominance is not universal, but then law of segregation is universal. Okay, wherein law of dominance, what are the clear statements that are passed out? Characters or traits occur in factors, each factor occurs in pairs and each pair will have two contrasting alleles. Dominant allele will inhibit the effect of recessive allele. This is all about law of dominance. In law of segregation, it is clearly said that in a single factor or gene, when two heterozygous alleles are present, they will not blend or mix with each other. They stand with their unity or they stand with their individual identity. They don't mix or blend with each other. This is what said in law of segregation. And law of segregation is universal. Law of dominance is not universal. Clear till here? Any doubts anywhere, ma? And then, yeah, about test cross we have learned. Now, what are the exceptions of Mendel's laws of inheritance, especially for law of dominance? According to law of dominance, the dominant allele will mask the effect of recessive allele, yes or no? But if you take the concept of incomplete dominance, that is proved by taking flower color from dog flower or snapdragon. when flower color is considered from the plants, it is commonly known as dog flower, from snapdragon or antirrhinum. Okay, so when two flower colors are considered, red color flower when it is crossed with white color flower, homozygous red crossed with homozygous white. In F1 generation, you obtain all the pink flowers with heterozygous genotype, which means the dominant allele here is not completely masking the effect of recessive allele. That is the reason both the effects are shown here. This is not because of mix or blend of these two alleles. Both the alleles are expressing its phenotypes and dominant allele is not masking the effect of the recessive allele. That is the reason in F1 generation you obtain all the pink flowers. In F2 generation, if you see, in F2 generation, dominant alleles and recessive alleles, dominant and recessive alleles, when on selfing of F1 generation, in a Punnett square you can clearly observe, one will be dominant phenotype, two will be of intermediate phenotype and one is recessive. This intermediate phenotype is proving out the concept of incomplete dominance, and this is against the law of dominance which is given in Mendel's pattern of inheritance. And here in incomplete dominance, both the genotypic ratio and phenotypic ratio, genotypic ratio and phenotypic ratio is 1:2:1. Clear till here? Any doubt anywhere, ma? And then codominance. Codominance is usually observed in blood grouping. Codominance is usually observed in blood grouping and lentil seeds. First we will understand about lentil seeds once. Lentil seeds, two type of seeds were taken. One seeds were spotted seeds and one seeds were dotted seeds. On crossing this in F1 generation, what is obtained is both spotted and dotted seeds were obtained. Fine, right? So this spotted and dotted seeds are again showing out the phenomenon of what? Incomplete spotted and dotted seeds when both are expressed. This is again showing the codominance concept. This is showing out codominance here, like spotted and dotted, both the phenotypes are expressed in F1 generation. When this is selfed, okay, you can get 1:2:1 ratio of 1 spotted, 2 spotted and dotted and 1 dotted seeds. This ratio is obtained on selfing of F1 generation, which is codominance, where both the phenotypes are expressed in the F1 generation. Previously, when flower color is considered in incomplete dominance concept, two phenotypes are not expressed. Dominant allele is not completely masking the effect of recessive allele, so the color range is reduced from red to pink. But then recessive allele's phenotype is not expressed. Nowhere white is observed in the flower. Okay, it is completely a pink flower. That is how you can differentiate between incomplete dominance and codominance. Okay, so this is the concept of codominance. In our next class, we will learn out the example of ABO blood grouping, followed by multipluralism, pleiotropy, and then we will also look into dihybrid cross. Okay.